All right. Hello, everyone. My name is Matt Olson. I am a community relations manager here for the U.S. MEF2C Foundation. Welcome. It's great to see so many people here. Out of curiosity, raise of hands, how many were here the last time we did this in October of 2024? Good amount of hands. Who's new? Who's new? Great. Great to see that. Regardless if you are returning or new, I really hope that this weekend, this conference does provide some sort of value to you. There's a lot of great information from the people presenting. As you could tell, the community here is very important to us, and getting connected with the community is very important, so hopefully there's some sort of value there. For agenda for today, give a brief overview there. We have 5 presenters. Each time they'll come up in about 20 minutes presenting up on the screen here, and then about 10 minutes of question and answer. If you do have question and answers, we do have postcards at the table that you could bring up to myself, or we could bring the mic around to you. The food, the buffet will be in the back. That will be coming out about 6 p.m. We ask that we can get the kids served first and then adults after. We have a pre-recorded presentation coming out around about 6:30, 6:35, so that'd be a good time for adults to start eating and then enjoy the presentation at that time. A brief background about myself and my daughter, Tegan. That's what brought us here. So my wife, Kelly, and I were from Minnesota. We have 3 daughters. Our youngest, Tegan, has MEF2C, a partial deletion. She was diagnosed with a partial deletion when she was 1.5 years. Like most of you, all of you, a lot of uncertainty, a lot of worry, being scared was a very natural and immediate feelings that came about us. What we ended up doing was we really sided on the side of positivity, and we decided how can we get involved? How can we get it to a community that can help support us, that we can support them? That's when we found the U.S. MEF2C Foundation, got connected with Ann, and right away we were trying to find out how can we get involved? How can we provide value? How can we learn? The very first thing that we tried to do was we did a volleyball fundraiser. We've done it 3 years in a row now. It's a lot of fun. It's a great way for us to connect with our local family and friends and connect with them on what's going through with Tegan's journey and provide updates with what we're involved with here. So it's great to provide updates to family and friends on what's going on in the research world, and also give us hope on some sort of treatment for our little ones in the future. With that, community was very important to us on getting involved. It's very evident in the past 2 evenings, getting to know several people that we met before, and also new families, that just seeing parents get connected with other parents that have kiddos that are going through similar things, and being able to listen, being able to share, see it in, I see it and I feel it in other parents on being able to say, hey, there's starting to be a connection there. And I'll speak for Kelly and myself. We truly believe that no parent should be doing this alone. So being here, starting to be connected with a community is very powerful. So I hope, again, community can be a source for you all as you're going through this with your kids. Special thanks to the older children, especially from, my daughter's only 4. Special thanks to the parents with the older children, kind of paving the way and showing us on things that have worked well, have not worked well, and it has us learn even faster. So I know it's harder the older they get, so it's very appreciative on our end. It is also very motivating for us to continue to be involved in, because there's going to be other kids that are going to be younger that can learn from what we've gone through. So we really appreciate that for all the parents that have older children around here. Lastly, thanks to the U.S. MEF2C team that helped set this up. There's a lot of effort in there. Anne, Michelle, Tushar, Rachel, Laura, Judy, Kelly, thank you. There's a lot here. I think everyone there, please, round of applause. A lot of effort went into getting this set up. Also, thanks to Dr. Chung being a strong advocate for our group. A lot of connections there with Anne and continuing to be a strong advocate for us. So thank you. So first speaker this evening is Dr. Wendy Chung. Dr. Chung is Principal Investigator of Simon Searchlight. She is also the Chief of the Department of Pediatrics at Boston Children's Hospital. Thank you.
Okay. So I want to give you the headlines for those of you who have heard me talk about MEF2C before. Nothing to panic about. There's nothing that's going to be radically different from anything that I've said before. And for the researchers that are here, also to realize that any of the resources that are available through Simon Searchlight are available to the research community broadly, so that if any of you have friends that you want to bring in, we want to make your investment into the science be able to go to as many people as possible. So I'm going to very briefly run through this. But in going forward, as I was saying, one of the things that's I'll talk about, it's a little bit complicated, is that everyone is special. And I really do mean that in all sincerity. When it comes to the genetics especially, although everyone's united here as being part of MEF2C, individual children, individual people have slightly different genetic variants. And we're still, and as we're doing this, we have ideas about how it may affect MEF2C function, but I won't say that we've got it all figured out. So it's incredibly important for everyone to be represented for themselves, so that as we go along and as we figure this out, we'll be able to make sure that we leave no one behind. So I mean that in all sincerity, and it is really important to have yourself represented as we go forward. There's more complexity that, although there's a lot that's MEF2C, there are other factors as well, and at least my observation from what's been going on in the community as I watch you is that we learn from each other. And in a very good way, we're learning tips and tricks from each other that are helping in terms of being able to avoid some of the speed bumps that are coming along the way. So I encourage you, whether it's over dinner, whether it's some of the casual conversations, but let's put our heads together in terms of the things that are very practical today, in terms of the tips and tricks we can learn from each other going forward. The other take-home message that I'll give you is that I actually have a lot of excitement in what is going on around the world in terms of whether we call it artificial intelligence or other things in terms of going on. I think of it as assisted intelligence. And so there are a lot of technologies that are being developed that are helping in multiple ways. For the researchers here, it's helping us to be able to do more with the limited amount of time and funding we have, so that we're getting more out of everything that we're doing. We're able to see patterns earlier, we're able to connect the dots better. And so this is definitely something helping science move forward faster. But I also hope that it's helping our kiddos. And what I mean by that is that there may be ways, and again, this comes in terms of the tips and tricks, that help in terms of being able to support our young people to be able to be the best that they can be. And so as you're doing it, I also urge you to think about and learn from others. And they don't have to be others with MEF2C. They can be other children that have some sort of challenges, but being able to figure out which of those technologies are helpful, whether it's assisted communication, whether it's other things in terms of being able to just get through the day or the night. But there are other technologies that I'm watching you all in terms of families to be able to deal with this. And I'm sort of keeping my own personal list in terms of tips and tricks that are working, but I think it's going to be helpful in terms of helping our kids as they're growing up. So the point that I want to make is the Simon Searchlight team is a powerful army of people behind you. This is solely sponsored by the Simons Foundation. And in going through it includes all of the people you're seeing here, some of whom are in the room and many more of whom are back at home, being able to make all of this happen. I'm not going to go through tons of detail, except to say that it really has been over the last 15 years that we've been working to make this happen for you and for lots of other people around the world. And again, this is mostly for the researchers to be able to get access to any of the de-identified information. So all of your identifiers, all of your name, where you live, your date of birth, all of that stripped out of the information to protect you in terms of your identity as a family. But then it's available to all of the researchers, whether they're halfway around the world, whether they're in a private company. We just want to make sure we're bringing good things to the scientists doing it. This is a large community, includes not only MEF2C, but about 180 other neurogenetic conditions. And as we do this, it's not just one and done, but it's actually over the life course because we want to see how things change. There are critical times over the life course, like adolescence is one of those times. And so we need to understand what are those particular things that are changing. I apologize because I know we ask you to do a lot of things in terms of the different online surveys. Some of them are medical, most of them are about development, about brain development and epilepsy in particular, as I'll get back to. And as we do this, we ask you to do it over and over, not to just be pains in the butt, but because we really want to see, especially if you're learning something that works and being able to see if we're seeing a consistent pattern across the community. So as we're doing this, we do try and verify the information in there. I won't go through all of the details. You're going to hear from Misha at the end about how we do this from a genetic point of view, but we also do things with in-person collection, as many of you did today as well. And that helps to, in part, verify what you're telling us and to be able to get correlates of what you're seeing with things that you couldn't know. So things that we can see either inside the body or by doing direct examinations. So as we're going forward, I'm just going to go quickly through this. We do also, I'll say, this is being recorded so that people who aren't here today can be watching it over time at home, but we also give these as distributions. We call them snapshots in terms of what's going on so that you don't have to wait once a year to see what's going on. And if this is one important component for the research community, if there should be clinical trials, and you're going to hear a little bit about this from Sid next, but if there are things that would be treatments, this is a way to be able to let you know about it if researchers decide to use this way of contacting you. It's kind of being a matchmaker in the middle and letting researchers that might have other things they want to do to prepare for clinical trials or the actual clinical trials themselves. And so we have this as a, again, resource for the community to be able to use to put the pieces together. Okay, I'm going to go relatively quickly through so we can get to the heart of this. As we're doing this, we put the guidelines out for your doctors to be able to know what they should do to take care of your kiddos. We make that freely available and we update it over time. And that helps your doctors very, very quickly. When you're in the emergency room, they've never heard about MEF2C before, be able to quickly go up on Jane Reviews, get a really quick snapshot in terms of what it is, and be able to know how to help and work with you as you're going forward. Okay, so I'm going to go very quickly to what we have. So Misha's going to go over this a little bit later, and any of the researchers who are interested in this particular slide, I'm glad to send it to you. But these are the genetic variations we have in the MEF2C community. Each one of these dots is representing a person, and so you'll see that in certain positions we have a kind of a pile-up, or it's kind of a not in a bad way, but we have a lot of people that have variations at the same positions. There are other places where we have only one single person, at least within the registry now, with that particular variation. And that's what I meant when I said that it's important that everyone is represented, because we don't know how much of the differences we're seeing between people is due to this slide, differences between the individual genetic variation that we're seeing in MEF2C versus whether it might be something else. Now, some of the researchers I can see looking at this, and I can try and read their minds, they're trying to think through themselves, are all of these different genetic variations acting in the same way, what they might be doing to differences in terms of how that protein is functioning. And I think we're going to hear some of that later today as we go through, and you're happy in the Q&A to ask us questions or to sidebar us. But this is just a visualization and an example of how I want to try and make sure you can see the same things that we're seeing. Here's another way to visualize the data. Here we are around the world. We're not just here in Boston, we're not just here in the United States, we in fact are around the world. And this condition, I think, is equal opportunity. That is that I don't think this is something that happens to be an American condition or European condition. I think it's probably equally common around the world. It's just a question of if people are getting genetic testing to be able to find out their diagnosis equally around the world. What we are finding, though, is in a good way, people are being diagnosed more frequently and earlier, so we're seeing more people, both in Simon Searchlight as well as other places. And in the United States specifically, since we're in the United States, you can see where we are around the country, but there are many more people who are not on this slide, who I'm sure are out there, who are certainly welcome to join the party. Okay. I'm not going to go into too much detail, but again, for the researchers, any induced pluripotential stem cells are available through the Simons Foundation, and happy to talk to you about that later. But let me go to the real heart of this. So we have limitations in the information we have. So I wanted to clear that right at the front. This is showing you the age distribution of who we have in the Simon Searchlight. In a good way, we have a reasonable number of people, over 50 people now who are represented, but we still have largely young people. So we still have people largely under the age of 20. Again, it's not that I don't think they're out there, people who are 40s, 50s, 60s, but they might not have had, in fact, probably haven't had the genetic testing to identify the diagnosis, but they're critically important. And if any of you, based on connections you have in your community, they hold the key to us in part for what to expect in the future, to the extent that you can think about how to connect with people who are a little bit older, who might be those beacons of light to help us understand what to expect in the future. This is probably one of the near-term things that I wish we could do to be able to get some of that insight that will help us know what things to avoid in the future. I don't forecast for you all that there's anything disastrous coming, but I will say for other conditions, it's been very helpful for me to have that information, knowing some of those things that were coming. And I'll just give you an example. And I'm not saying that this is what you're going to have to worry about. But if your child had appendicitis, it would be very likely that it would take us a little bit longer to diagnose the appendicitis because they might not tell us, I've got pain right down here and it's hurting me, and be able to really articulate it and say that. Any delays in diagnosis for us as doctors tend to be associated with complications. Your appendix could burst before we could know that we need to treat it, before we need to take it out, or before we need to give antibiotics. And so in general, if we know what to look for together, then we can anticipate those problems, watch for those problems, and hopefully get ahead of those problems. And so that's an example. And again, it's not that appendicitis, I'm worried, is going to be happening to everyone. But in general, when we know if it might be screening for heart disease or diabetes or cancer or whatever it is that's going to come up for our kiddos as adults, longer term, I want you guys to be prepared. So that's one of the limitations we have for the data we have now. It's not going to surprise you. None of this, I don't think, is going to surprise you. You're Dr. Mom and Dad or Grandma and Grandpa in terms of doing this, so you could teach me more than I can teach you. But what we know is that this is largely what I consider something that's above the shoulders. In other words, a brain condition, a neurological condition. But in some cases, the brain is connecting to other parts of the body. So we shouldn't be surprised that we might see gastrointestinal issues, for example, because we know there are brain cells that are going to the intestinal tract. We see the problems that you all know about, so none of these are surprising. And we do have some individuals who have autism diagnoses as an example as part of this. And as part of that, hopefully they're getting the services they need in schools and in their support systems to be able to help them with that. Besides neurological issues, though, we see some other issues that are important. Vision is important. If you can't see, it's harder to be able to navigate your world, and vision is one of the things that we see more frequently. I know you guys know that, but especially as we have little ones coming into the community, it's important to be able to keep that on our radar. Seizures are another important issue, and you'll hear also from some others today about this. In a good way, for most people, they aren't overwhelming, but they are something that we need to be able to pay attention to so that if we see the seizure, we can treat the seizure. And if you have any questions, go back to your doctors, your pediatrician, your neurologist in terms of bringing up the question, and they can easily do an electroencephalogram to look at it more frequently. Next up on the list in terms of frequency are those gastrointestinal issues that I was talking about. And again, there are nerve cells that are going to the intestine. Sometimes we see that the intestine isn't moving things quite along quite as quickly. So we see things like constipation. Other times it's moving things along very quickly. So occasionally we'll see diarrhea. And sometimes the muscles aren't quite so strong. So we see things coming back up in the form of reflux and heartburn. Good news, there are medications that help with a lot of these things. There are diets that we can use for a lot of these things. But one of the problems is sometimes I don't think we realize when someone's not having a great day, they're having an off day, sometimes it's tummy trouble. Sometimes it's things going on inside that, again, they can't necessarily tell us about as easily. And so consistency is one of the things that's quite helpful. When you find a formula, a combination, something that works, stick with it is my advice. Consistency in terms of that. And again, that's where the tips and tricks come in helpful, is being able to find out from a dietary point of view, from a regularity point of view, what are some of those tips and tricks to use. We generally see problems in terms of muscle tone. You're probably going to hear about that more from other people as well. As we see that, it can lead to some of the problems where some folks need braces from an orthopedic point of view to be able to help with some of the hyper or hypotonia and some of the things that we can see, especially during adolescence. For those of you who haven't reached that time yet, kids are going through their growth spurt. Sometimes we're seeing a little bit more in terms of curvature of the spine or scoliosis. And again, something to keep an eye on with your pediatrician. If your orthopedic surgeon needs to be brought in, they can, but something long-term in terms of keeping folks healthy. And then some of the other things, thankfully, are not nearly as frequent. In fact, I would argue probably no more frequent than the general population. But sometimes we have picky eaters, and so they're not gaining weight so much. And sometimes as we're doing, they may not grow to be quite as tall, but nothing that's, I think, overly concerning or that needs a lot of treatment. These are just getting into some of the more, the things that the neurologist may be noticing and being able to share with you. Some of these are, like I said, different words for different things I've said. Low muscle tone or hypotonia is one of the ones being common. Issues in terms of coordination, where it just takes a lot longer to learn how to be able to do something, but there is muscle memory, and practice makes perfect in terms of going forward. But learning some of that coordination, whether it's coordination with the fingers and the fine motor skills or coordinations with the big body movements, they take a lot more in terms of patience to be able to get to the point where we need to get, but they'll oftentimes get there. This is one of the ones I was alluding to before in terms of seizures. So we do see seizures, not in everyone, thankfully. As we're thinking about these are different types of seizures, so they can look differently to you. In some cases, they might be big jerking movements. In some cases, they might be kind of just looking off to the side. It might be a little bit of eye twitching or mouth twitching. But this is saying there are different types of seizure types that we see. And within even one person, there can be different types of seizures at different times of the life. And so, again, if there's a concern, go to your neurologist with this. For vision issues, some of these are things that you can see in terms of just looking straight on at the person because their eyes aren't lining up exactly straight. And so you know, and that's an easy one to tell. Some of them, it's a little bit harder to tell whether or not they're seeing things well from a distance. But a good pediatric ophthalmologist is the key in terms of being able to help you with that. And either whether it's a surgery that's necessary or glasses that are necessary, but can help to make sure that we can navigate the world. I'm not going to go too much into this, but again, as I said, in terms of growth issues, a little bit is what I would say, not too big. The orthopedic issues I talked a little bit about before. That curvature of the spine is another way of saying scoliosis. And then another thing that people find helpful is what conditions are serious enough to need medication. So just being able to look by frequency, you can see that one of the other things I hadn't talked about was sleep. So one of the other things that can be an issue is sleeping. And sleeping, I would say, is contagious within the family, in that sometimes if a child doesn't sleep well, all of a sudden there are other people in the family that don't sleep so well either. And so for me, this is an important thing to be able to help. Melatonin can help, other stronger medications can help as well. And so these are some very specific examples of some of those medications. I'm not endorsing that that medication is a medication that your child should specifically use, but sometimes, you know, when you go home, your doctors may say, is there anything you learned from the conference? Feel free to show a picture of this slide, or we're happy to give you copies of the slides themselves. Okay, I'm going to very quickly just go through in terms of what we've seen with I screwed it up. Go back. So these are some of just the summary in terms of what we've seen overall. I will say in terms of development, things continue to go forward. We haven't seen a point where things are going backwards, so I don't see this as a degenerative condition, but that slope in terms of how much and how fast things go forward, I know how slow that can be for many of your kiddos and how much repetition and diligence it's taking. But the good news is we're still not seeing that this is a degenerative condition. My hope is that it isn't. So I wanted to relay that and to say that, anyway, we will move on to Sid. Or I'll let Matt introduce the next speaker, I should say. Or questions. Do we take questions? We take
Questions, yes.
Okay.
Before we do that, we do invite the presenters to sit up at front because then they can be involved in the Q&A as well. First question that we have, what are the variants we've seen in the MEF2C community? Do you think variant diversity, different functional outcomes will lead to the disease diversity evidenced in research? I think that's posed towards Dr. Chung and potentially Dr. Aiken.
For those of you who remember, there are a lot of different genetic variants in the gene, and I'll pose this to my colleagues as well. I think in certain cases, here it is, I think in certain cases we think that some of these genetic variants, we have a pretty good sense that they cluster together in terms of how they function. I won't claim that we know every single one of them in terms of mechanism of action. I actually think it's one of the things that some of these folks or in their laboratories are working on. So I'll invite my colleagues to join in.
Yeah, I will be touching upon this question in my presentation in just a little bit, but I think, spoiler alert, I don't know if there's, the strongest evidence right now is that there are most definitely mutations acting through haploinsufficiency, which is a big fancy word that means that one copy of MEF2C isn't enough for, for health of a neuron or for health of a person. But based off of a lot of the things that Wendy just beautifully went over, including all of the different positions where mutations can happen, in addition to the wide spectrum of disease, one of the things I'm really dedicated to looking at in my lab is to see if these distinct mutations do give rise to different functional changes within a cell.
Next question that we have. When we look at MEF2C point mutations, some result in frame shift mutations, which we see throughout the gene. Others result in nonsense mutations found in the functional domains in the last exons of the gene. And others result in missense mutations in the MADS box region. Why do we see only nonsense mutations in the functional domains in the last exons of MEF2C and missense mutations in the MADS box region?
Wow, we're really getting into genetics here. I love it. So I'm not sure how familiar everyone in the room is with these different types of mutations, but we just kind of got a little, I can give a little crash course. So nonsense mutations will introduce a premature stop, which means that you only get part of your protein. A frame shift means that the DNA kind of gets squished so that when you make your protein, it will look different than what it's supposed to be. And typically that leads to it not being functional. And then a missense mutation is when you have a single amino acid change. So just like you have your protein structure mostly, and then just one little change. And I think that the question that Matt just asked is really interesting. And, I could speculate, but I think that there's something interesting about that MADS box domain, which binds to DNA, that it's either there or it's not there, or you get missense mutations. I don't know. I feel like there's a lot to discover still about how these distinct mutations alter MEF2C.
Whether there's a protein made, you kept on saying that the point mutation, truncated protein, frame shift, something truncated as well. Whether a protein is made or not is an open question, and certainly it may well depend on where the mutation is in the gene. Right.
Yeah, as well as nonsense mediated decay. There's all kinds of things that can regulate and lead to a loss of function ultimately.
All right, next question. Is there a relationship to the type of variant, deletion versus mutation, and seizure activity?
So the good news is that there's no group that's having terrible, terrible seizures as a whole group. So we do see a little bit of variation, but I would describe MEF2C in general as not, there are some conditions, some neurological conditions that are primarily epileptic encephalopathies. I don't really think of MEF2C as one of those. There are a little bit of variations from person to person as we're going forward, but I wouldn't say any class as a whole is a severe seizure disorder.
Is there any questions from anyone in the audience? I'm happy to run the microphone to you.
My daughter has a 5q14.3 deletion, so she's missing, I don't know if I'm talking really loudly, she's missing MEF2C, RASA1, and 10 other genes that don't seem to do much, thankfully. In your experience, does the 5q14.3 present a lot differently, or I assume most of it's being driven by MEF2C, but is the fact that it's fully deleted along with other genes maybe more severe? Or I see that question come up a lot on the Facebook group and there doesn't seem to be a clear answer.
Yeah, so I'm happy to talk offline for individuals who have individual level questions. What I'll try and broaden this to is the generic question, which is that most people have just MEF2C that's involved, and in many cases, it's just one single letter that's changed. There are a few people in the community that have not just MEF2C, but it's kind of MEF2C and its neighbors next door and down the street. In general, the thinking is that if you have more genes involved, there's a greater probability that some of those other genes will do something in terms of the way the body's working. Not always, not every gene, but sort of size matters, and bigger is unfortunately not better in this case. And so in general, and we can go offline, you have to inventory the exact genes that are involved. You can't just say it's a 5q deletion. You have to get down to the nitty-gritty of which genes are involved, and we can go through that and inventory which genes, sort of which ones matter and if it's something that does matter.
Any other questions? All right. We will go to the next presenter, Dr. Siddharth Srivastava. He is the pediatric neurologist at Boston Children's Hospital. Thank you, Dr. Chung.
Excellent. So thank you so much for the invitation to talk here today. I'm really delighted to talk about some of the lessons that I've learned from clinical trials for cognition and behavior in different genetic neurodevelopmental disorders. I'm going to start this talk by talking about my journey up until this point, because I think that's really paved the way for the subject of this talk. I like thinking about food, so I'll illustrate my career journey accordingly. I grew up in Baton Rouge, Louisiana. I did my undergraduate in Columbia in New York. I went to medical school at Hopkins in Baltimore, where I did my residency in child neurology and neurodevelopmental disabilities. And I spent as much of the time there as I could in the neurogenetics clinic there, which really solidified my passion for this patient population. And then in 2017, I moved to Boston, where I did a fellowship in neurogenetics and have stayed on as faculty since then. At Boston Children's Hospital, I provide care to children with a wide range of different neurogenetic and neurodevelopmental presentations. I co-lead our section within of neurogenetics and neurodevelopment. And within this section, I see patients with a multitude of different genetic neurodevelopmental disorders, including Phelan-McDermid syndrome, P10 syndrome, Kleischer syndrome, and many others. And I also evaluate patients with cerebral palsy, where one of my niches is to try to figure out what are some genetic conditions presenting as cerebral palsy. My overall mission is to basically try to improve the lives of children with neurodevelopmental and neurogenetic conditions by trying to translate scientific discoveries into meaningful clinical practice changes. On the clinical side, I'm focused on providing comprehensive care to individuals with genetic NDDs and trying to optimize long-term neurodevelopmental outcomes. And on the research side, I'm really focused on trying to help develop targeted treatments for these conditions through natural history studies, as well as targeted treatments for many different genetic NDDs. So in this talk, basically what I wanted to do is I wanted to highlight some of our work on a few of these different genetic NDDs, trying to go through some examples of some of the natural history studies we have done, some of the clinical trials that we've done as well, and try to talk about some of the valuable lessons that we've learned from these conditions. So I'll start by talking about one example, which is a syndrome called Kleischer syndrome, which is a rare genetic cause of autism and intellectual disability. A few years ago, we started a clinical and research program for this condition through collaboration with the patient advocacy group. And after a couple of years, we created a multidisciplinary clinic and have seen 100, 110 patients since then. Fascinatingly, as a subset of these children have this period of neuropsychiatric collapse that happens in adolescence, and that starts off with really bad insomnia, that's followed by neuropsychiatric symptoms, that's followed by regression in self-help skills. And this raises a lot of really interesting questions about the underlying gene, which is EHMT1. How does it impact sleep? How does it impact regression or loss of skills? And if we better understood the relationship between sleep disturbance, is that a key to understanding the relationship with regression or loss of self-help skills in this syndrome? And so on the path to trying to unravel some of these questions, we connected with a neurochronobiologist named Jonathan Lipton, and he's really focused on studying molecular mechanisms involved in circadian rhythm, which is our body's way of generating sleep and wake cycles. And we started talking about Kleefstra syndrome as an example and became really interested in this condition. And since then, we've been doing some really interesting work, basically showing basic science work that basically shows that this gene involved in Kleefstra syndrome, how important it is for regulating circadian rhythm. And specifically circadian amplitude, which is important for the robustness of your internal biological clock, which can relate to a lot of really important processes, including metabolism, how our cells process and use energy, as well as a multitude of other processes. And at the same time, I connected with a person named Joe Kosowski, who's doing work with biometric recordings of sleep, so using things like Fitbit devices and whatnot to track physical activity and sleep. And so his lab was using these various devices to try to assess sleep metrics like sleep staging and heart rate and physical activity. So it made sense for all of us to partner together, and that's basically what we did. And what we essentially established is this cross-disciplinary research paradigm that involves quantitative neurodevelopmental phenotyping combined with biometric recording of sleep, combined with understanding the basic science, the biology of this underlying gene. And more specifically, we've been collecting some neurobehavioral data, including a measure on repetitive behaviors called the RBSR, a measure on other sort of maladaptive behaviors called ABC. We've been collecting data on adaptive skills using an instrument called the Vineland, and data on communication using an instrument called the Orca. We've been collecting data on sleep using some standardized assessments, and participants enrolled in our study have been wearing these Fitbit devices for 1 to 2 weeks at a time. We've also collected some skin fibroblasts, some skin biopsies from some of these participants, looking at the biology of specific patient mutations. We're looking at the data right now and coming up actually with some really interesting connections that are tying all of these different domains together. And importantly, this work, again, this data has been single time point, but it has laid the groundwork for us to start a natural history for this condition, which we launched a few months ago and have already reached our target enrollment. This natural history study builds on this cross-sectional data, on this research paradigm that involves the intersection of all of those key components. So I think the key point of all of this, like one of my core lessons to you guys, to the whole group at large, is from this whole experience is that it's been so fantastic to build these cross-disciplinary connections and collaborations that can lead to better understanding the mechanisms, mechanistic insight, that can lead to really interesting and cool new avenues for investigation. It's been very exciting to do so. And by the way, I just really like the idea of impacting clinical practice on a big national kind of level. So one of the things that we've been doing is to try to spearhead the creation of an ICD-10 code for Kleefstra syndrome through our work with the CDC. And this might seem trivial, but this, I think, is a really important thing to do and something to consider, because this can lead to identification of patient cohorts and for the purpose of ongoing and future research, trials, and studies. And so, again, think about doing so for the MEF2C group at large. I want to talk a little bit about a second example, which is Phelan-McDermott syndrome, which is another genetic neurodevelopmental disorder that's caused by a defect in a gene called SHANK3, which is a synaptic protein. Not surprisingly, kids with this genetic syndrome can have a wide range of neurodevelopmental challenges, autism, intellectual disability, behavioral challenges, neuropsychiatric symptoms, seizures, psychiatric conditions, and so on. And I've been lucky to be part of this ongoing consortium called the Developmental Synaptopathies Consortium, which is headed by Dr. Mustafa Sahin. This is an ongoing NIH-funded natural history study that's looking at Phelan-McDermid syndrome, P10 syndrome, which I'll also talk about, and some others conditions. And this has been going on for 10 years, and we've gotten funding for additional 5 more years of data collection. The study has involved a wide swath of different sort of neurobehavioral assessments pertaining to cognitive, adaptive, which are self-help skills, communication outcomes, motor outcomes, autism symptoms, other sort of symptoms, and importantly, various biomarkers, including EEG biomarkers. And I wanted to focus on one of the instruments that we've been collecting data on, which is the Vineland, which is a measure of self-help skills. This is a standardized assessment that looks at self-help skills in terms of communication, in terms of socialization, and in terms of daily living skills. And for each of these big major domains, there are also these subdomain scores, receptive, expressive, written under communication, interpersonal, play and leisure, and coping under socialization, and personal, domestic, and community under daily living skills. And this is getting a little bit into the weeds, but bear with me for a second. For each of these big, large domains, such as communication, socialization, and daily living skills, you can have a standard score, which is a score that's normalized relative to a general population of same-age peers. But you can also have these so-called growth scale value scores, which are scores that measure change relative to your individual, that individual's previous progress. And so the first point I want to make is that when we started looking at the data, again, this is getting a little in the weeds, but I'll try to get to the point, which is that there's a large percentage of our cohort that we're basically bottoming out in terms of this measure, that we're basically scoring just at the very minimal of each of several of these measures. And again, we wanted to look at changes in these adaptive scores over time. So I'll skip over this. This is just a slide just showing the methodology we use. But importantly, I'll walk you through this slide. This is a slide that looks at how have these scores changed over time. If you look at estimate and if you look at the number, a positive number means that there's progress. A negative number means that it's opposite of progress. And what I've highlighted here is that, again, see for communication, 2 of the subdomain growth scale values are showing progress, or showing change over time, which is good, but the standard score hasn't changed. That's interesting. And then same story with the daily living skills. Some of the growth scale value scores are showing change over time, but the actual standard score is not showing change over time. And then for socialization, the growth scale value scores are not statistically showing changes over time, and the socialization standard score is actually looking negative. It's looking like it's going down over time. So at this point, you should be saying, well, wait a minute, does this mean that this population is regressing or going down over time? And the answer is that not really. It's just that the individuals are making such small progress over time that the standard score is looking like it's going down. It's just that the growth scale value, but there's just minimal or very little progress that's being made over time. And again, with respect to cross-sectional effects of age, so looking at all the individuals kind of at once and seeing effects of age, basically we found that older participants tend to have lower Skinner scores or being more affected than younger participants. So what are all the takeaways from this? What are some of the lessons from this? I think first of all, when you look at the data cross-sectionally, meaning not longitudinally, but just all the time points at once, older individuals are more affected than younger individuals, which actually parallels data from other genetic NDDs and from other cohorts of patients with intellectual disability and autism more broadly. And then within a given individual, so there are some mild increases in adaptive skills in certain areas, but I think it's really important to note that in some cases, some of these changes over time are not sufficient to impact the standard score and actually make it look like it's going down. So the key point of all this is that when doing the analysis of the data, it's really important to look at these so-called growth scale values, at least with respect to the Vineland, an adaptive measure, because that can show that there's actually incremental change happening Because if you just look at the standard scores, you could be misled in thinking, wait a minute, this is a regressive condition or something's going on that isn't actually the truth. So the main takeaway is that there's a great need for looking at these gross scale values along with the standard score to try to interpret change, especially when you're dealing with the population that can be low functioning. And this data has been, or these lessons have been used in an ongoing clinical trial. We're doing a gene therapy trial for this condition. So the last thing I just wanted to, last point example I'll mention is from a syndrome called P10 syndrome, which again is a genetic NDD that's associated with a wide range of challenges. You know, more beyond the kind of granular or coarse kind of assessment of a high prevalence of autism and intellectual disability, one of the things that we've looked at is trying to see what is the granular neuropsychological profile of the syndrome. And again, I won't go into the weeds with the slide, but the key point is that it's really important to sometimes get down into the nitty-gritty of the neuropsych profile, not just the full-scale IQ, but the attention and the processing speed and the working memory and all of those kinds of things. Because for this syndrome, we actually highlighted this interesting profile that we said is attributed to these, characteristic of frontal lobe systems. But on a practical level, this was helpful when I was talking to a patient recently with P10 syndrome in an adolescent, and we actually used this data to help get them actually more time on standardized testing, because we said, hey, this is a syndrome that's associated with working memory, processing speed deficits, and I think extra time is needed. So that's just an example of using research data on a very practical level. Yeah. And then so we just mechanistically, we know that P10, this is a gene that impacts a pathway called the mTOR pathway, which plays a role in cell growth. And so what we did is we did a clinical trial using a drug called everolimus, which is an mTOR blocker. So in P10 syndrome, there's too much mTOR activity. So we used a drug that blocks that mTOR pathway down, trying to target cognition and behavior in this syndrome. And this is work that was led by Dr. Hardin in Stanford. This is a clinical trial that we did, multi-center trial. We enrolled about 50 participants, and half were in the placebo arm and half were in the treatment arm. Again, this is a busy slide, but the main takeaway is, so our primary measure was this neuropsych composite of working memory and kind of more of a reaction processing speed and a motor coordination assessments. And our primary outcome measures didn't show improvement. So initially we were disappointed, but then we looked at some of the secondary measures, including nonverbal IQ, that showed some change, some improvement, tendency for improvement. We also looked at one of our social measures, the SRS-2, that showed a statistically significant improvement in the treatment arm compared to the placebo arm, as well as some of the motor coordination tasks that we did. And this is just a graph that shows that there were differences between those who received the treatment versus the placebo in these motor coordination measures in the social measure as well, which was very exciting to us. We also looked at EEG biomarker data. Again, main takeaway is that we showed that there were changes, neurophysiological changes as evident by the EEG signatures that we did, suggesting that there's target engagement, that the medication is actually working on a very neural circuitry mechanism level, which is very exciting to us as well. And again, this is the main kind of takeaways is that although our primary endpoint was successful, because we cast a broad net, and this is really important when you're thinking about clinical trials, casting a wide net and doing a wide range of assessments can be really important and can try to highlight signal that might not have otherwise been revealed. And we used EEG biomarkers that showed evidence of target engagement, meaning that the brain was being impacted with this medication. And this has paved the way for another trial, which we're about to launch using this drug, but targeting social communication, because that was one of the measures that was actually that showed signal with our first clinical trial. And again, as I was mentioned earlier, it's really important, I think, to think about clinical practice changes, applying this knowledge in the form of patient care. So trying to build clinical practice guidelines that can be applied to, that can be used by other physicians and other patients is really important. So lastly, just to conclude, so some of the key takeaways here are that these cross-disciplinary collaborations are so important, I think, and it's so exciting to me. With natural history studies, especially if you are working with the population that can be more affected cognitively, I think it's really important to look at not just standard scores for some of these assessments, but look for growth skill value scores if that instrument allows it. And then with outcome measures for clinical trials, sometimes it can be hard to find a signal with the primary endpoint, and sometimes you need to cast a wide net to try to detect signal in those trials. And then finally, it's so important to try to aspire for clinical practice changes in the form of guideline development and so forth. And with that, I'll just end my talk there. Thank you.
The first question that we have, do you have advice, feedback on clinical trial design based on experience with other gene groups?
So there's lots of clinical trials that are going on in this space across multiple rare genetic disorders. We have a lot of information about what the FDA is looking for. It changes over time, but we're really able to stay quite up to date. But the main important thing is that you need to have a clinical trial before you can refer to those prior trials to see which ones are most appropriate. But there are tons of things going on in this space, and the FDA has been really, really fantastic of late about working with the family foundations, understanding what's important to the families, trying to adapt the studies in such a way that they are minimally burdensome and also address the concerns of families. So we have a well-trodden path at this point once this group comes into clinical trials.
There are two things. One is trial design, and if it's something where you can do it, the most rigorous way to do it is what we call double-blind placebo control. So what we mean by that is a way where both you as the family don't know what someone's getting, and we as the doctors don't know what's getting. So none of us is biased in terms of our observations. A lot of the trials we've been doing recently do that as the first part of the exposure or taking a pill. But the second part is open label, so that everyone has the chance to be able to get exposed to the medication. And during that phase, the family knows and we know, and so we can see what's going on, but it gives everyone the opportunity to be able to be exposed. That's good and bad, because these are experimental. We don't know if these medications are going to do good things or not. Within those, it keeps both the rigor of the study, in my opinion, as well as being able to have everyone have a chance to be able to get exposure. Some of the new types of trials that might come forward, though, are things like genetic therapies, where I don't know that we're going to have the same rules in terms of randomization. Many of those are done as open label. That is, everyone knows that they're getting a genetic therapy in those cases. And that allows people to make more informed decisions because there's higher risk and there may be one chance in terms of doing those. But my greater concern, which Sid was all of Sid's talk, was how are we going to know if this, whatever it is having an effect? And I do worry that being able to see the change, a lot of the standardized measures that we use can't see small changes that are important to you, and we're going to have to work on that now before we get to the clinical trial. And so that's what a lot of people are working on, is we call it clinical trial readiness, being prepared, so that as soon as we do have something for a trial, like, we're ready to go. So that's a lot of what you've been about here today for those of you who have done the in-person studies. We thank you for your patience of that, but it's really trying to do kind of a practice run and figure out what will be part of the clinical trials of the future.
That's great. Any questions from the audience?
So the question for Dr. Srivastava, my question is, in your clinical trials, did you find or get any example where there is an adverse side effect or, very like something is deteriorated very badly? Any example do you have, or did you
Sure.
experience?
Absolutely. I think whenever we're talking about an intervention, whether that is a FDA-approved medication or whether that's an intervention that's a research basis, that's a research intervention, there's always going to be a potential for adverse effects. As part of the clinical trial design, that's something that we monitor very closely. In fact, some of these trials start off with the primary focus is on safety first and foremost, followed by efficacy. So to give you just some examples for some of the drug trials, again, this is not going to be specific to MEF2C, so this is just some examples from other genetic conditions, and each therapy will have its own kind of set of side effects, but sometimes there can be non-specific side effects such as GI side effects, upset stomach, nausea, appetite changes. In terms of as you're going to more invasive types of therapies, for example, for Phelan-McDermid syndrome, we're doing a gene therapy trial right now. That gene therapy trial involves injection of a medicine directly into the brain. So that is a high-risk procedure, and the highest risk is potentially even death in some cases, or that's a theoretical possibility with gene therapy trials and has happened in some instances as well. So to answer your question, there can be a wide range of possible side effects from minimal to quite severe. It really depends on the specific intervention, specific specific medication that is being used.
Thank you all so much for being here. And I'm sorry I came up and said hi. You probably would have made the rounds here. What is the feasibility or viability of doing a qualitative portion to any of these studies?
So what we do in a lot of these is that I actually think you guys are the best observers. You know your children the best, and you can see a change, whether it's a good change or a bad thing, but you can see the change. So if that's what you mean by qualitative in terms of being able to journal or keep a note, or increasingly, I do videos where individuals will videotape before and then during. And so if they're seeing something, literally, we can see it as well. We've had things where I will admit sometimes the first trial it's not perfect because we didn't realize what the drug would do. And so then we get a hint, though, of something that might be happening, or drug or other treatments. And so I think the more that you guys can be careful observers and document it in a way that is a way for us to be able to go and see what you're seeing. And that's why I like videotaping or audio recordings. I think we learn during that process. And for me, it's how can we sort of cycle faster to be able to go from learnings at one stage to then something that ultimately is successful later. So that's in the things that we do, we build that in.
Any other questions? All right. We will thank you, Dr. Srivastava. Next up is Dr. Jane Aiken. Dr. Aiken is Assistant Professor of Biomedical Sciences at the Colorado State University.
Well, thank you so much for inviting me here. I'm really looking forward to getting to know some more of you all during dinner. But I'm, as he mentioned, an assistant professor. I'm just now getting my research team started, and I'm really excited to be able to build it from the ground up, centering around some really important questions that I think we've identified in MEF2C research. So a huge thank you to Anne for really bringing me on early. So today I get to talk to you about MEF2C variants and how there's the potential for them acting via different mechanisms. So here's just an outline of some of the things I'll get to talk to you about today. We're going to dive into some cell biology about MEF2C function, looking at the identification of MEF2C variants. I'll provide an example of some of the research that I've done in the past on a different gene group and using iPSCs for disease modeling. And then finally, link it all back together for our future plans for MEF2C research. So to start up, here's a little Cell Bio 101. I myself am a cell biologist at heart. I love coming here and interacting with all of these MDs who have this amazing clinical knowledge that I am lacking, but what I do have is a very robust knowledge of how cells work, which I find very fun. And I really love to use the analogy of cells being built by a bunch of proteins working together.
And in this analogy, I like using Lego bricks as proteins. Yeah. And so you don't just need one protein and put them together. You actually need a lot of different proteins that do different things at different times, and they all work together to make a specific cell type. And so in this example, one cell would be Bob the Builder. But if you had different proteins working together, you could get different cell types, such as Pokemon Geodude. You might notice from my analogies I have a 5-year-old son, but I particularly like the analogy of Geodude as a neuron because neurons have to extend these long processes or like arms that link themselves to form these synaptic networks, which will come up later in my talk. So within our DNA, our genome, all of the instructions exist for all these different types of proteins. And so how does a cell know which set of instructions to follow to get the right proteins? And one of the, really the major way that it knows how to do that is by using combinations of transcription factors, which you may have come across the term of because MEF2C is 1. So if you have a combination, say set 1, you could get all the proteins you need for one type of cell, and if you have a different combinations, you would make another cell. And so if we were to zoom in on this DNA, you could actually see MEF2C performing its major role, which is binding to specific DNA regions in order to help regulate expression of other genes. So dictating which instructions will be followed to make those proteins. And in particular for this talk, and I think really in general, we're really interested in the role of MEF2C in regulating neuron function. And as I mentioned earlier, neurons are all throughout the nervous system, and they have to link together in order to send messages to allow us to think, to talk, to move, and so many other vital functions. That brings me to the end of my little spiel about what MEF2C is doing, and then getting to the identification of disease variants in this important gene. So the first and that brings me here. As we all know in this room, mutations to MEF2C can lead to a wide spectrum of neurological symptoms, which Dr. Chung beautifully outlined for us in her presentation. It's actually just fairly recently, if we think about the timeline of our understanding of these things, that MEF2C was recognized as a disease-causing gene. And actually, the first identification was of one of the deletions, a microdeletion of the chromosome 5. That was the first report involving MEF2C. And it was really just about a year later when we recognized that MEF2C specifically, if you just do MEF2C, not any other genes in that region, MEF2C was enough to give rise to these pathogenic conditions. That same year, they found that not only deletions, but specific amino acid changes or mutations can give rise. There's still this idea that it's going to be mostly a loss of function. These early stages really point to a number of these mutations causing either deletion or loss of function of MEF2C. But what I find really interesting is 3 years later, we've actually identified that duplications to MEF2C can actually cause pathology, which suggests to me that changing MEF2C levels within a cell can cause problems. And I don't think that as a field, we really understand all the ramifications of how changing its function will lead to severe neuronal challenges. And that's this big question that I'm really trying to step in and start addressing in my lab from the cell biology perspective to understand how these different mutations, because if I go to an openly available clinical database, there have been over 600 reported variants in MEF2C. Dr. Chung showed up that there are, I think I was counting, there's around 35 specific amino acids that can be affected. Here's a kind of out-of-date schematic that I was able to pull off of another publication. And more importantly is that when we actually look at our patients, we're seeing differences in how they develop and how their neurological system is able to function. And so that brings me to an example of the type of research that I do using a different gene but that mirrors MEF2C in a number of important ways. And so in this example, I look at KIF1A, which is a different gene within the cell that expresses a protein that's important for moving things around the cell. And so if you remember with a neuron, that can be really important because a lot of the things are made in the cell body and they have to be sent out to those long arms. So moving them around at the right time is very important. And like MEF2C, with KIF1A, you can have a number of different disease-causing variants all throughout the protein. And I was able to rely on a very robust data set of literature, part of which was generated in the Chung lab, to take those nearly 200 different mutations and classify them based on how they are either predicted or shown to change the way that motor can move around the cell. And in this specific case, we found that in some cases there are mutations that would lead to haploinsufficiency or not enough of that protein. Other times they will lead to a partial loss of function. It's doing its job but just not as well. And other times, which I think are really interesting, it actually leads to a gain of function. So these motors are moving too fast, or they're binding to, or they're getting stuck and not able to go at all. And we were able to partner with Jackson Labs to help create a really important resource in understanding how those different changes to that motor protein's function could result in different changes at the neuron level, at the network level, and then ultimately reconcile those with what we're seeing with patients. And so this resource is a line of iPSC cells. So iPSCs are induced pluripotent stem cells. Feel like they've kind of had their day, and you may have heard of this term. But basically what these are cells that we have made to look like stem cells, which gives them the capacity to turn into any cell that your body needs. And in this particular case, we're interested in turning them into neuronal cells because that's where MEF2C seems to be doing most of its work. Or in MEF2C and also KIF1A. There's a lot of parallels here. And this is what this looks like in an actual experiment. We take our iPSCs and we can differentiate them into human neurons that we're able to perform experiments on. And if we look at these distinct changes to the motor function, we find that they have different outcomes on how these cells develop and behave. So in this particular assay, we're looking at how the neurons are able to extend those long arms that link to each other. And you can see that different mutations cause different changes relative to the wild type or normal condition. We're also really interested in how this is changing things at a functional level. So a neuron's main function is to form synapses and send those synaptic messages. And we're able to look at how well neurons are able to do this using multi-electrode array electrophysiology, which is a mouthful, but basically we grow the neurons on these arrays and they record spontaneous sending of those messages within the network. We'll see if this plays. Oh yeah, it does, which is really exciting. So I, being a cell biologist, was very excited when I learned about this technology because it gives us beautiful access to like high throughput and longitudinal data regarding how these cells function. And what we find when we look at this for these specific different types of KIF1A mutations, we find that different mutations can give really different outcomes in terms of synaptic connectivity. So in fact, the one that causes the motor to go too fast actually leads to an acceleration in neuronal connectivity, but then declines. We think potentially due to excitotoxicity, so it's sending out too much stuff and the neurons are like, overstimulated, whereas the other mutations, either the haploinsufficient or the partial loss of function, leads to a more delayed, compared to this black line you see, which is the normal condition, delayed synaptic development. And so this data really suggests that potentially for these, what it really shows is that distinct variants within the same gene, within the protein, can give very different outcomes from the molecular level, at the cellular level. And that also potentially it might be really valuable to look at tailored therapeutic strategies based on what's going on in the cell because different things are happening. And so to finally wrap up my talk, I want to bring it back to MEF2C, linking what we know about what it does, the types of strategies that we've used for other genes, and think about what we might be able to do to understand MEF2C in different variants. And so just to bring it back, a protein needs help figuring out what proteins to express. Transcription factors help regulate which genes are going to express and which proteins will be inside the cell. And so, what really brought me or made me very interested in this particular gene is the fact that when I look at the specific genes that MEF2C is helping turn on, a lot of them fall into this category. I've color-coded them green, of being really important for transporting things throughout a cell, which is what KIF1A is important for and a number of other things that I've studied throughout my research career. And so my lab is now well equipped with the tools and strategies to look at the cell biology when we make these changes to MEF2C and see how it's altering neuronal trafficking and neuronal function. And so, and really get to this question here about potential disease mechanism and disease mechanisms, where you might have different things happening depending on the specific variant to MEF2C. In particular, we're interested in probing how these variants alter MEF2C's ability to bind to specific regions of the DNA. Also, what does that mean downstream for the overall gene and protein landscape of a cell? And then linking what we understand there to how it's functioning at a cellular and tissue level, using iPSC-derived either neurons, like I showed you for the KIF1A story, or in organoids or mini brains, where we can make the iPSCs recapitulate early stages of brain development. And then finally, we want to integrate these insights that we're getting across scales, from molecular to network level, back to the really important patient data that's being collected by Simon's Searchlight to understand how these different changes to MEF2C ultimately lead to neurological symptoms. And with that, I will just leave you with my thanks and take any questions.
Any questions from the audience to start with? We do have one that we had before, so I think you alluded to what your lab was doing now. This question: how will your investigations lead to treatments for MEF2C related disorder?
So one of the things that I'm really excited about with our multi-electrode array strategy is that we can look at more than one condition at a time. We actually have 24 different wells that we can manipulate. So this system is really exciting from the perspective of you have a lot of different ways to read what's happening to a neuron. So you can add different compounds, you could do different genetic strategies, and read out in a pretty high throughput, rapid way what interventions are making the MEF2C cells look more like the baseline wild type cells. And this is all very, basic science preclinical data, but I think it's going to be really important for informing some of these, clinical trial, ideas downstream. And that's why I think it's so nice to have these interdisciplinary meetings where we can see what everyone's doing and help inform, future therapeutics.
Any questions from the audience? Great. Thank you, Dr. Aiken.
Thank you.
All right, next up is Dr. Sandeep Datta. Dr. Datta is from the Harvard Medical School, professor of neurobiology.
Awesome, great, terrific. Hey everyone, my name's Bob. I am a neurobiologist. I work just across the street. Thank you so much for including me in your day. This talk's gonna be weird for a couple reasons. So the first reason it's gonna be weird is this is basically, I'm a basic biologist, and this is a technology talk. So I'm going to tell you about some technologies that my lab has been developing over the past decade to understand behavior in general, behavior largely in mice, but we're increasingly, as I'll show you, applying those technologies to humans. The second thing that's weird about this talk is that I really want to apply this technology to MEF2C, and we haven't done it yet. So at the end, it'll be obvious how that will be applicable. And at the end, I'll tell you about our plans for doing that. All right. So a lot of what my lab is concerned with is trying to understand how neurodevelopmental disorder-related genes affect brain circuits and then ultimately behavior. And so just as you heard earlier today in discussions about clinical trials, how challenging it is to know what to measure, to figure out whether a drug is working or not, it's super challenging to actually understand how mutation in a gene changes the behavior of something as simple as a mouse. And so we do really simple, in some sense, experiments in my lab. So this isn't a typical experiment. In this experiment, I went to Home Depot and I bought a bucket. You can see the bucket there. And then I got a webcam, like you'd put in your door, and I put it on top of the bucket. And then I bought a mouse, and I threw the mouse into the bucket. And when you do that, this is what the mouse does. So the mouse doesn't just run around like crazy, and the mouse doesn't do nothing. Instead, the mouse actually explores the bucket. And through careful neurobiological and neurobehavioral analysis, we can actually prove that what the mouse is doing right now is building a map of this bucket. And the way it does it is really interesting. It moves its body through these micro movements that you can see. So like the mouse will pause and sniff a little bit. It'll turn, it'll rear. Sometimes it'll stop and groom itself, which is what it's doing right there. And through these organized movements that evolve over time, like a language, like a mouse body language, the mouse is able to understand the world around it. Okay? Now, so my lab has really sought to try to understand this body language because we figure that the brain builds this body language and it's the way the animal actually interacts with the world, right? And one thing you'll notice about this very simple movie is that mice aren't two-dimensional, even though this movie's two-dimensional. Mice are 3D, and a lot of their movements are actually up and down. And so we wanted to figure out a way to measure the 3D behavior of this mouse. So it turns out that up the street in Fenway, there used to be a Best Buy, and I sent a graduate student to that Best Buy, and he bought a video gaming console called the Microsoft Kinect, which actually includes in it a 3D camera. So it was a camera that you would use to monitor your own movements as you played a video game. And Alex just took that camera and swapped out that camera, that 3D camera, for this 2D camera. And when he did it, this is the movie he got. Okay, so here you can see the mouse, and you see the mouse is now colored, and the color in each pixel actually represents the height of the mouse at any given point. When the back of this mouse is red, it's arched up towards the camera. You'll occasionally see the nose turning red, just like right there. That's when the mouse is reared up, high, kind of sniffing in the sky. All right? So now we have this beautiful data describing how mice in 3D explore a little bucket. And our idea was to try to understand what this mouse's body language is actually kind of built out of. And so we did a pretty simple thing, which is just we used some simple video extraction technology to extract the image of the mouse from the bucket and to align it, as you see here on the left, so that its nose is on the right and its tail is on the left, okay? So that movie you're seeing on the left, that red movie, is the same as the movie on the previous slide, okay? So it's a mouse running around in a bucket in 3D. We then took those pixels, the pixels that make up, the little dots that make up that image, and we just laid them out on the y-axis of this graph and played that movie forward. So the movie on the left is the movie on the right. They're the same exact thing. They're time-locked, okay? But what you notice in the movie on the right is that the mouse seems like it's stuck in a pose, and then there are these lines that separate the behavior of this animal over time into these blocks. And we thought that each of these little blocks might actually be a kind of unit of behavior. And when we looked at movies of mice and aligned them to the data, what we saw was that indeed, each one of these little blocks was a repeatedly used little action the mouse generated, like scrunching up or rearing or running. And so we decided to call these little units of behavior, which tend to last a couple hundred milliseconds long, behavioral syllables, because we thought they were components of mouse body language. And we wanted to discover somehow, through some means, the set of behavioral syllables that an animal expresses in a given environment, and the behavioral grammar that governs how those syllables are strung together over time, right? So you want to know both what the parts of behavior are and how they're organized over time to let the animal meaningfully kind of explore the world. And so we took advantage of artificial intelligence to build an algorithm that we call motion sequencing, or MoSeq, that takes movies in 3D movies like you're seeing here on the left, and from those movies automatically identifies all of the different types of behaviors the mouse generates and tells us the order in which they occur over time. In other words, it's an artificial intelligence platform that decodes mouse body language, okay? Now that's a lot of data, and it's sometimes hard to work with a lot of data, and so we can compress all these data into a single simple visualization, which is shown here on the right. So on the right, each one of those circles is a behavioral syllable. So maybe, let's see, you see that? That one over there would be rearing, that one over there would be sniffing, that would be running, okay? And the diameter of those nodes is how often that syllable occurred in a given experiment. And importantly, the thickness of the lines that interconnect these syllables depict the likelihood that one syllable will follow the next. So these lines depict basically the order in which these syllables occur. And so this representation has two important properties. The first is that it lets us do a really amazing thing, which is to predict what a mouse is going to do in the future based upon what it's doing now. Because we know that if the mouse is doing that thing, the next thing it's going to do is this thing, and then it's going to do this thing, then it's going to do one of these two things. So that's pretty cool. But the second thing it gives us is an objective and quantitative summary of every behavior the animal generated in this experiment. And what that means is that when we make mutations in genes that are related to neurodevelopmental disorders that change an animal's behavior, we can use changes in this map to objectively identify the ways in which behavior has changed. I want to mention one other thing that we've done with this technology that's really important for our basic understanding of how genes relates to circuits and behavior, which is that we've rendered this technology compatible with all of the modern, currently available forms of neural recording. And so we can simultaneously record brain activity as we let an animal run around freely to understand how this behavior relates to brain activity. And I'll actually show you an example of this. So here in this movie, this 3D movie here in the upper left, is exactly what I showed you before. It's a mouse running around in a bucket. You'll notice a big difference about this movie. This mouse is wearing a hat. Out of that hat is coming a cable. And that is because I've implanted into this mouse's brain a miniature microscope that's visualizing in real time the activity of neurons in the frontal cortex of this animal. On the right is a movie from that microscope. So what you're looking down at here is actually the mouse's brain. Each one of these dots is an individual brain cell that's involved in processing information. And when there's a flash in that cell, it means that neuron is currently active or communicating with other neurons. And we can then, post hoc, use motion sequencing to identify which behavioral syllables the animal is expressing and look at the activity of each individual neuron in this part of the brain and relate these two things to understand how brain activity gives rise to behavior. And through a careful analysis of these kinds of data, we've been able to identify key neural circuits involved in the construction of natural self-generated behavior in the mouse. Okay, so how does this help us think about therapies? So if we want to think about therapies and disorders and diseases, we have to ask ourselves whether or not we can usefully use this kind of method to understand how drugs might influence behavior. We want to ask how these kinds of technologies might capture changes in behavior caused by differences in genes, like changes in MEF2C. We want to ask about whether we can identify interactions between those two things. So let's take drugs first, okay? So my lab has done an experiment in which we've taken basically every known neurally active drug that you can buy commercially, and we've given those drugs to mice and thrown them in buckets and filmed them. And so we've generated a data set of about 2,000 different mice using maybe 60 different drugs at about 5 or 6 different doses each. And we've simply asked whether or not motion sequencing, this technology, can identify all of the changes in behavior that are caused by treating animals with these 60 drugs at these different doses. And so one way to think about that is whether or not we can use differences in the syllables that are identified by motion sequencing to tell drugs apart. I'll skip ahead. I'll go back. This is something called a confusion matrix. You don't have to understand it. All you have to know is that if I can predict which drug a mouse is on based upon its behavior alone, there should be white along this line. And And what you can see is that this motion sequencing technology lets us, just by looking at a mouse running around in a bucket, know exactly which drugs it's been treated with, and it allows us to know the drug and the dose. Okay, so this technology is really valuable for telling us the ways in which behavior is changed by a given drug. And that means we can make maps of drugs. And so each dot here is a drug, and the position of each dot in this map reflects the different patterns of behavior elicited by each of these drugs. And so the drugs over here in this map are stimulants: methamphetamine, modafinil, methylphenidate, which is what you'd expect. Drugs over here are antipsychotics, risperidone, haloperidol. And this kind of map is really useful and important because it allows us to take new drugs and to ask how they're likely to influence behavior. Okay, so we can also look not just at drugs, but at genes. And so we've been looking quite extensively in the lab at mice that harbor mutations in autism spectrum disorder genes. And so here are two genes that are known in humans to cause autism. One's called catnap2, and the other one's a deletion in 16p11.2. And we know that these mutations of these genes in humans and in mice make children and mice hyperactive. If you measure how often mice that are mutated in these genes run around, they run around more. And if we use our 3D cameras to measure whether mice are running around in a bucket, you'll see that mice of both of these different mutations run around more. So this is the mutant and this is the wild type, okay? Given these data, we wondered whether or not we could tell apart a mutant animal and a wild type animal using motion sequencing. And so it turns out that you can. So this is a very simple plot called a usage plot. The y-axis is just how often each syllable is used. Each of the tick marks on the x-axis is a different individual syllable. And you see there's a bunch of syllables here that are used more in the catnap animal when compared to these white dots, which is just a wild type animal. And it turns out each of these dots is a running or a darting or an exploring behavior. And so the increase in these specific behaviors explains why these mice are hyperactive. Now, we've tested a bunch of different genes that are implicated in neurodevelopmental disorders. And so we have a pattern of syllables that's changed that's unique to each one of those. And so if you gave me, say, 3 or 4 different types of mice, catnap2 mouse, a mouse deleted in a gene called SHANK3, a mouse deleted in 16p11.2, just by looking at the mouse with this technology, I could, in 5 minutes, tell you which mutation the mouse had. So there are really significant differences that separate these different gene effects. And as I told you before, I can build a map of the different syllables that are changed with drugs. And so in this example, we know that these syllables are upregulated in the catnap mutant. And so we went back to our drug map and we asked, are there any drugs that make these syllables actually go down? And maybe if we gave that drug to a catnap mouse, its behavior would be normal. So if we do that experiment, and we could find a drug called loxapine, which has never been tested before, as far as I know, for the treatment of hyperactivity in the context of neurodevelopmental disorders. And you can see that it actually works. So in these circles are syllables that previously were elevated in the catnap animal that are now normal. The X's are syllables that weren't fixed by loxapine. And the square is a syllable that was previously normal that is now abnormal because the animal got loxapine. So this is a side effect. And so what this tells us is that for any given gene that's mutated, we can identify a kind of signature or fingerprint of the ways in which their behavior has changed. And we can very precisely ask how candidate drugs might change, might ameliorate specific phenotypes associated with that mutation. I also just want to mention something that came up earlier that Wendy had mentioned, which is that all patients are individuals. And we have very poor ways of thinking about individuality in the lab in mice. And it turns out that, and that's really important because you want to model not just the gene, but also kind of individual effects related to a given gene. But it turns out that this motion sequencing technology actually captures individual variation in mouse behavior. So in this experiment here, each dot is an individual mouse, and the position of each dot represents the mouse's pattern of syllable use on a given session. And here we just took a bunch of mice and we ran them in the bucket for seven straight days. and we made this plot. And what you see is that this mouse, which is colored with this color, always has the same pattern of syllable use in the bucket, and it's different from this mouse, which always has the same pattern of syllable use, which is different from this mouse, which also always has the same pattern of syllable use. And so what this means is that mice, like people, have personalities. They're all individuals, and they all stably are themselves, and because they each have personalities, they're distinguishable. I can just look down at a bucket and tell which of 16 mice I was looking at in that video, just by looking at the specific pattern of syllable use the mouse generates. And so this tool is going to let us think about individual variation in disease as well. We've begun using these tools to analyze human data. So this is actually accelerometer data taken from an ASD subject that is having a repetitive movement. And we applied motion sequencing to these data to try to identify behavioral syllables that make up a repetitive motion. And you can see here actually the algorithm learning what the syllables are. And you can see it does a really great job of automatically identifying the different components of a repetitive movement. And so we're excited about using this kind of technology to analyze the kind of Fitbit and other kind of data that you've heard about earlier today, and to get a real sense of the ways in which behavior is structured in folks with developmental disorders. Finally, there's a lot of facial data that's available in this setting, and we can accurately use this technology to identify facial syllables. This is actually a subject whose hand is on a hot plate, and so she's grimacing, and so we can identify pain-related syllables quite effectively, but also just behavioral syllables. And it turns out that this technology, spookily enough, works really well as a lie detector. If we talk to her, we can tell when she's lying, we can tell when she's telling the truth. We can actually do a terrific job of predicting how bad her pain is from these syllables. So, and because these data are relatively easy to obtain in humans, we think they're going to be this technology is going to be applicable to humans on short time scales. So there's just to end, Mike Greenberg and I currently have a project to develop a new kind of MEF2C model mouse. That mouse is under development. And our plan is to use this technique to understand both brain changes over time, over development, and behavioral changes over time, to really get a rich sense of how mutation of MEF2C might alter neural circuits and alter behavior. And then by design, this mouse is essentially optimized to allow us to rescue or to restore MEF2C expression in particular brain regions at particular times that we choose. And that'll let us ask if we can somehow develop a therapy that replaces MEF2C, say in the forebrain, early in development, will it restore behavior, back to normal? And so these are experiments that are currently underway. We're obviously kind of early in the process, but I expect if you invited me back in 2 years, I'd have something useful to say about this. And with that, I will just end, and thank you for your attention.
Any questions from the audience? We had some here. First one I'll share. How is reversibility of disease in mice comparable to reversibility of disease in humans? How are the similar biological characteristics? Or excuse me, what are the similar biological characteristics?
So I think we're still working on reversibility of. So I think we're still working on reversibility in mice. So it's too early to answer that question. But I think the technologies I'm describing to you here are going to play an important role in really capturing phenotypes both in mice and in humans, and might provide us with a kind of common language for understanding the ways in which reversions and therapies might be related to each other, like the ways in which things are fixed might be related in mice and in humans. And I do think like these kinds of technologies, I'm actually going to a meeting just next week at the Simons Foundation to talk about these kinds of technologies and their application to human subjects, because I think this is really the future of finding better and richer ways to phenotype folks with different disorders in order to understand whether clinical trials are working and to understand the ways in which patients are individualized, to understand the ways in which therapies might vary. I mean, I think this is really, really crucial. And I think this kind of approach overall is going to be an important part of that conversation.
Another one here. How can AI be used in drug discovery for MEF2C? And your experience with AI changing over time and how that's helped your research.
Yeah, so when I started my lab, I never imagined that I would use any of these tools, much less develop any of these tools. It's really transformed our ability to understand the structure of the brain and how it relates to behavior. I mean, I will say that the brain is the most complicated machine in the known universe. I think there are really good reasons that we don't understand it. Not just the numerosity of the neurons, but the complexity with which they're interconnected and the complicated dynamics with which they talk to each other, right? And these AI tools are allowing us to make sense of all that. And so in some really interesting way, at this moment in neuroscience, our ability to collect data, whether it's from behavior or from neural circuits, as I showed you, or from patients, our ability to get data is actually much better than our ability to understand the data. And AI is really helping us get clarity on those kinds of issues.
Yeah, is there a finite number of syllables, or are syllables sort of like an emerging property depending on environment, et cetera?
Yeah. So the type of algorithm we use is what's called, this is a little bit in the weeds, but it's called an unsupervised algorithm, which means that it discovers syllables depending on their use, which means that there's not a finite number of syllables. Instead, if you imagine if you put me in a restaurant, the syllables of behavior I would generate would be different than if I were sitting in a symphony hall, would be different if I were playing football. Same thing with a mouse. And so this technology will just discover whatever pattern behavior you generate and tell you how it's organized.
I'm not sure if it is a correct question or not, but at what stage right now we are in terms of MEF2C?
Yeah, so not very far at all. We're just starting working on these mice. So people have previously generated mice that are deficient in MEF2C. As you heard earlier, there's lots of different mutations associated with MEF2C, and there's actually quite a bit of debate as to whether the existing mice do a good job of capturing what's actually happening. And so in terms of understanding MEF2C related behaviors, I think in mice we're just at the beginning. And certainly in terms of applying these kinds of tools to humans with MEF2C deficiency, we're also right at the beginning. But I think it's an exciting time to begin to think about applying these tools both to mice and to humans.
Any other questions? Thank you, Dr. Dada.
Thanks much.
All right, our next presenter is Dr. Joseph Buxbaum. He's also here with Dr. Iyer. Dr. Buxbaum is a professor of psychiatry, genetics, and genomic sciences at neuroscience, and he wants me to just skip all that. He's being bashful. Thank you.
So it's considered a very, very bad idea to stand between the audience and their meal. It's multiplied several times because we have both adults and children here, and so standing between children and their meal can be deadly. I will try and kind of hit the high points. Please interrupt me. I know this is tough stuff. I didn't understand the prior lectures at all, so it must be hard for people who are outside the field. I'll talk. Shruti wanted me to talk so she could watch. She could laugh at me later, but I'll explain her role in this as well. We're from the Sieber Autism Center at Mount Sinai, which is a big center of about 60 people working on rare genetic disorders for a big part of what we do. So, there's a new concept that came out a couple of years ago called profound autism. So we say we work on autism. This much better term for what we work on really is profound autism. It's autism that's defined by, kind of functionally by, needing lifelong access to an adult, right? Probably can't live independently. And, the other thing that's also called is IDD, intellectual and developmental disability. And, this focus on profound autism and IDD has really changed things because people now realize that it is a fairly significant problem. We are not alone. And I say, every family here, but also my own family and other families are affected by these sorts of disorders. And the concerns tend to be, like the top concerns of families tend to be, what you all know about, communication challenges, cognitive and adaptive challenges, getting older, being independent and all those sorts of things. And obviously there are neurological concerns like epilepsy, and safety and so on. And we do some things for that, and we can do them better, right? Support and services needs and so on. But what we really want to get to is called disease-modifying treatments, treatments that modify the disease, don't just touch the symptoms, which is of course important, but actually change the trajectory. And the really amazing news, and why I think it's one of the most exciting times to be working in this space at any time, is that there have been a couple of trials recently over the past 2 years that have shown very compelling changes in these central issues, right? So this is Liz Berry-Kravitz talking about some trials that are going on in Angelman syndrome, which is a genetic disorder as well. And what you can see here is improvements in communication and daily living skills in this example here. And there are also cognitive changes as well. And just, well, now last year, 3 companies have been given FDA approval to do a phase 3 trial in this disorder with this gene-targeted therapy. So there's a lot of optimism that some of these things that many people kind of treated as maybe, how can we change them? How can we change communication and cognition and daily living skills? That they actually may be changeable. And this is another study. Again, Liz is involved in it. This is a study of a phosphodiesterase drug in Fragile X syndrome. It was done both in children and in adults, and it too is now in a pivotal trial. And what you can see is improvements in cognition, in language, and in here, this is the Vineland you heard about earlier, in daily living skills as well. So, you know, this is all in the past couple of years where we begin to see that we can actually make changes in these kind of central developmental issues, right? And even in older kids and adults, like in the Fragile X trial. And we're all pretty sure, actually, that this drug over here, which is not specific necessarily to Fragile X, we could argue that, may actually help more than people with Fragile X. It might be a general cognitive enhancer. So that's why we are really, really at a point now where gene-targeted therapeutics is really at the forefront of everything we do. We're very excited about it. It's also a bummer that this is the time where, who remembers Marky Mark and the Funky Bunch? Okay, so Marky Mark is another name, well, that's what I call the people who in our government right now who are making decisions around these sorts of things. So one of the things that we came to many, many years ago was it was quite clear that there were many, many genes that were causing these sorts of disorders. I was fortunate enough to put together a consortium that has been funded by the NIH, by your tax dollars now continuously since that time. And the whole point of this consortium was with lots and lots of genes, you can't just do it all by yourself. You have to make these massive data sets. And we've gone through every couple of years major papers. The parallel effort, one of several parallel efforts, but the one you've heard the most about probably is the Safari effort as well. And so, you know, side by side, this NIH-funded an effort and the Simons-funded effort have gone and found more and more genes. This is 2014. Many genes for autism, intellectual disability, developmental delay. This is a combination of the data sets. This is the 2020 paper. This is our 2022 paper. I just jumped ahead on. And we have these many genes that, when mutated, present with IDD, all the things that we are all familiar with. And so there are opportunities, if you will. First of all, and I can explain more in a moment, one of the things that became quite clear, and you've kind of already heard about this, is that as you began to look at this as a gene name, each gene, you began to see what kinds of mutations produce the phenotype, and you could begin to cluster them into those things you've heard about, haploinsufficiency, where missing some expression causes the disease, gain of function, other things like that. And so we can begin not only to understand that a gene is contributing to this disorder, but how it's doing it. And most of these are haploinsufficiencies. All right, and this is that word that you've already heard. Haploinsufficiency means having 50% of the gene product, because we have 2 copies of MEF2C, one from mom and one from dad. One of them is mutated, so you have half as much of the gene product is contributing to disease in a haploinsufficient condition. As you've also heard earlier, there is a thinking that most MEF2C, that there's certainly haploinsufficiency going on in MEF2C. There's probably more things going on, but that's certainly part of it, right? The other parts may be gain of function, dominant negative, something like that, where the mutated product, like the missense product at this side of the screen, is interacting differently with DNA, for example, and then changing the properties of the protein, right? So we can use our genetic data and then the functional data you've heard about as well to begin to map the mutations into haploinsufficiency or other things. That's a big deal, and I think we're well on our way with MEF2C from some of the paper presentations you've heard. Our most recent paper, with now 60,000 individuals with intellectual disability, developmental delay, and autism, and hundreds of thousands of controls, not only has, and many clinical samples as well, has MEF2C as one of the top genes. If you notice, this very small number of genes I'm showing you here out of the 500 or so that we know about now, maybe more, accounts for about half of the mutations we see clinically. So it's a really, even though there are hundreds of genes, there's a fairly modest number that account for most mutations. And my job in life is to convince people who look at this thing in biotech and pharma and other people like what we're doing on platforms to focus on the top genes, because those are the ones that are going to be most, we can make some relatively quick headway. So what do we do at the Seaver Center? We spend a lot of time trying to find genes for developmental delay syndromes, and then we try and make the iPSCs that you've heard about already. We also make mouse models, which you've also heard about already, and we do a lot of kind of the natural history stuff and phenotyping that Wendy and Sid do. The SHANK3 project that Sid was talking about started at Mount Sinai. We've been doing it for a long time, and we were very fortunate to be able to introduce it to the RDCRN when it became a national network. So, you find a gene, you now have a target, you can mutate that gene, or you can take human neurons, human nerve cells, and study them in a Petri dish. You can mutate that gene in a mouse, or a rat, or a dog, or a monkey, and you can understand how that affects brain development. And then you can also go back and look at the families and understand how that mutation manifests its phenotype. Sid talked about practice parameters. You can educate the families and the clinicians about what to expect. You can do, as Wendy said, you try and find people who are older and younger and get a sense of trajectory. And ultimately, you do a natural history study while you follow individuals over time, right? That's what you want to do. But what you really want to do is use all these tools to think about novel medications, novel treatments, and then put them in the family, in the individual to make improvements. All right. And so there's various ways to do it. And one of the ways that people are focusing on more and more is gene-targeted therapies. So the disorder is caused by a gene mutation. Can we actually do something to that gene to reverse the mutation or to compensate for the mutation, right? And that's, again, Sid talked about the study that's going on right now where the people are introducing in Phelan-McDermott syndrome, SHANK3, the gene, in an AAV within a viral vector into the brain with a lot of risk, with concerns for a lot of risk compared to what you took this morning when you had a little bit of headache because you were drinking last night, which is an oral small molecule therapy, right? And I know nobody here was drinking. So how do we do that? Well, first of all, how do we, at the Seaver Center, balance this? And you've kind of gotten it here, right? Some people are talking about mouse models. Some people are talking about stem cells. Some people are talking about, patient-based research. We try and do it all. We try and choose genes, and here are a few of them that we've chosen in detail. And we try, at the same time, seeing lots of patients, but also developing programs in stem cells and in animal models, and then kind of learning one from the other, going through this process of understanding the phenotype, understanding how the cells with mutations look different, how animals with mutations look different. Think about what that tells us about existing drugs that could be used for repurposing, but ultimately developing novel therapies that could be disease modifying. Right? And I won't talk much about iPSCs, but you've heard about them already. But these are kind of examples of iPSCs. And Trudy works with Nan Yang, who is one of our lead iPSC people in the program. So multiple labs are able to work on the same gene side by side, which is a lot of fun, actually. So I'm going to give you a couple of, I'm going to build up just one gene ADMP as an example of what can be done. If, one thing we're doing. Here is one of our papers showing ADMP as the top gene. Here's a more recent one. This is 2020, 2022. You see it's gene number 5 or something. And most of the mutations are PTVs, loss of function mutations. So we think it's a haploinsufficiency as well, although it too has some subtlety. And so, you know, what do? We've seen lots of patients, so we can take their bloods and we can look for blood-based biomarkers. We see a very, very profound difference in some blood measures in these individuals. Okay, so we have now a blood-based biomarker. So if somebody could make a change in that deficit, maybe the blood would show that. So we have a way of measuring that quickly. We've bring in all these families, just as you know, they come in for, what you've been doing, they come in for 2 or 3 days, they go through EEG batteries, they go through eye tracking, they go through many, many biomarkers and clinical endpoints. And this is an example where we have sensory measures. We can measure somebody's reactivity to sensory stimuli, even if they're nonverbal or preverbal, because we have experts, we developed a whole series of basically toys that have, that are tactile, or they have sound, or they have a kind of their visual stimuluses. And depending on how the child reacts to that, we can score it as a reactivity, hypo, or otherwise. And then we can, of course, do clinical trials, right? This was a clinical trial done at the behest of the families. So once you've seen a lot of families with a certain disorder, and you kind of have some sense of endpoints that Wendy was talking about, you can do clinical trials pretty quickly. So we've done all of this for ADMP as an example. So if you want to modify a disease, a genetic disease, right, you want a disease-modifying treatment, you can target the gene. In some ways, that's the easiest. You can also possibly target things that are downstream or upstream, and you certainly hope you can. But there's more and more technology and more tools to target the gene itself. And like I said, haploinsufficiency is the most common thing we're finding for a variety of reasons in these rare disorders. But it's also the case that we're finding gain of function and dominant negatives, even imprinting. ASO is Angelman, which is the one I mentioned earlier with the 3 clinical trials, is an imprinting disorder. But to the degree that some MEF2C mutations are gain of function / dominant negative, you want to maybe make a toxic product go away, or you may want to increase the expression of a gene where you have 2 copies, one has a mutation, one is intact. What if you can increase the expression of the wild type or the intact allele? And the most important thing about this, and we've already heard a little about it too, is that we're pretty sure that these genes are, their dosage sensitivity is what we call the Goldilocks effect. It was actually borrowed from NASA. But anyways, that too much or too little is bad, right? And you've already heard this mentioned earlier that many, many times haploinsufficient genes are shown to also be triplosensitive. So if we all have 2 copies of these genes. 1 copy, having only 1 functional copy is bad, but having 3 copies is also not great. And people are worried that some of the therapies we're approaching, what if we went up to, what if we pushed it and there were 4 times as much or 5 times as much? How detrimental would that be? There's unfortunately a reality in biology that biology is very much a U-shaped curve. There's an optimum, and then you can fall off the other side. And so dealing with this question of dosage sensitivity or the Goldilocks effect really is one of the reasons that there's a lot of concern about how we're going to move forward here. And I don't have an answer. You're welcome. But what I can say is that if we could use small molecules as opposed to drilling, putting a viral vector into the brain, which is what Sid talked about earlier, a small molecule is a traditional medicine that you take, by mouth. You have to take it every day, but you can also control the amount. You can go up very, very slowly. You can small amounts. It's non-invasive. Obviously, that's a good thing. It has much bigger safety profiles compared to viral-based interventions, and it's presumably reversible. And so the idea we're focusing on is this. We are looking for small molecules, traditional medicines, that will bind to part of the gene of interest, the gene that's mutated, and make it increase the expression. So there are two copies of MEF2C. One has a mutation that's non-functional. One doesn't have a mutation, it's intact. What if we can just push up the expression, the amount of protein that comes out of the intact allele? And so what we do is we take an enzyme from a firefly. Firefly? Who remembers Calvin and Hobbes trying to so the fireflies light up, and that enzyme turns out to be very, very useful, and it's been cloned out, and it's been put in cells in culture. So you can measure the amount of a protein, because that protein now has this it lights up, and so the more of that protein you have, the more it lights up. And we can put in parts of the gene of interest and connect it to the enzyme. And so if a small molecule binds to the gene of interest and makes more of it, we see more light coming from the firefly. And so then we can quickly screen, so far we've only screened 2,500 compounds at a time, but we have now a library of 10,000 we're putting together. Screen these small molecules chosen because they're drug-like and because they love to bind to RNA, and ask whether that small molecule has no change in the amount of light generated, decreases the amount of light generated, or increases the light generated. And what I'll talk about is increasing. So we're trying to get from, what was there to more, right? So this is called the reporter screen. And so, these are a series of small molecules, and these are wells with hundreds and hundreds of little wells and plates. And so you see most of them express a certain amount of luciferase, but a few of them express more. So the drug that's in that particular well with that particular dot is pushing up the expression of that gene product, right? And you can do controls and counter screens, and you can really kind of come to a point where you say, I have a small number, 33 in this case, of compounds, of traditional small molecules that seem to be pushing up the expression of ADMP in a reporter system. The next thing to do, and by the way, these are the collaborators, the next thing to do is to see whether that same drug increases the expression of the native endogenous protein in a living cell. So this is kind of washed out, so let's ignore that. These are two drugs that we found, P5G16, and as you put in more and more of that drug, 1 micromolar, 5, 10, 20, 40, you see that the amount of protein has gone up, right? Here's another drug, P1A9. And again, you can see that the levels of the protein go up. So we've done this screen with luciferase, with the firefly enzyme, and we found some drugs. The screen is very quick. We can do thousands of compounds. This is quite a bit slower, but we've taken those top compounds, and now we have 2 of them that actually can push up the amount of protein to where you want it to be. These are actually being done. This is also from Nan's lab. This is the same experiment now, and Shrita's working on these things, where we're trying to do the same thing. These are human neurons in culture. These are induced neurons, iPSCs. And what you can see is this is the wild type. This is a mutant, a patient-derived mutation. You see there's half as much protein as there was before. This is also a haploinsufficiency syndrome. And the drug works so well that you really, already at the lowest dose, you're seeing much more protein. This is the human neurons, human nerve cells being pushed up, the amount of this missing protein is being increased. Nan's lab is also looking at the iPSCs from the point of view of electrophysiology. You've heard about this earlier as well. And there are several very characteristic findings in terms of changes in the cell signaling between nerve cells when you have a mutation in this gene over here. And so we can use that as a physiological readout for the drugs. We don't have the results yet, but now we can say, okay, this drug we found in our reporter screen increases protein in mammalian cells, increases protein appropriately in human neurons in culture. Does it change? Does it reverse the deficit in cell signaling? Nerves exist to talk to each other, and when you have a mutation in this gene, the talking changes. And so will this drug reverse that communication deficit, cellular communication? We don't know yet, but that's what we're doing. And then once you have these drug-like molecules, you can begin to modify them very, very quickly, because this is what pharma's done for ages, right? You have a small molecule, you want to make it into a drug. First, you modify it so it becomes more and more potent. It works at lower and lower, it becomes more and more effective. You make sure it gets into the brain, right? So here are, a series of compounds, and for each one we have, we know how much it pushes up protein expression. We know at what concentration it does its job, so we can optimize very quickly. And we can even do something called structure-activity relationship, SAR, where we try many, many derivatives of those first effective compounds, and we ask what makes them less or more effective. These are about 50 compounds, and they cluster together. So this is one cluster, this is another one, and we can begin to optimize each one and move it forward pretty quickly to what's called a drug candidate, where we've taken that small molecule to the point where it is a drug candidate. It can be considered for your first trial in humans, first safety and then efficacy. And we're pretty excited about this because it's moved pretty quickly since we got it started. And me being excited about my own work shouldn't impress you, but we've gotten 2 fairly important grants in the past 2 months. This one is from Oxford Harrington, which is a rare disease foundation. And it's on exactly what I just showed you. So they said, we're really intrigued about this approach to, for this rare disorder. We'd like you to take it further. Another one will be announced, I think, pretty soon, but I'm asked not to talk about it. It's a fairly significant one as well. And when we started this whole thing, we picked, let me go one more. We picked 4 genes to start with. SHANK3 is the gene, one of the genes that Sue was talking about. ADMP I just talked about, DDX3X, FOXP1. And for all of them, we had some successes, right? So that's what's down here, right? And so we put those together and we presented them to the Simons Foundation last year. I'm going backwards again. Yeah. And the Simons Foundation said, we'd like to see whether you can stretch this out over many more genes, 10 more genes, right? So the Simons Foundation said, maybe this is what we call a platform. In other words, every time there's a gene of interest, maybe when you use this tool to relatively quickly come up with some hits to leads to drug candidates. And it's early days still, but we're pretty optimistic. We're running the next 10 now. I mentioned, and since that time, we've gotten the Oxford Harrington grant, another big grant that I think will be announced soon. And so we do think that, we're not the only ones who think about platform approaches, but a lot of groups are. And we think that this one has some advantages because it is oral, it is titerable, you can, increase things slowly. And if you stop giving the medication, the effect goes away. It's not like gene therapy where, it doesn't. So thank you from the center. And yeah, questions? Time for jumping jacks?
Questions from the audience? No, you're not that easy. We do have a few here. So, how can your investigations for other gene groups or on small molecular discovery be leveraged to address MEF2C deficiency?
Yeah, so the reason we've been focusing on sort of a platform approach is because I come from a group that discovered hundreds of genes, and we want to be effective and efficient as quickly as possible. So everything we're doing here is meant to be easily pivoted to another disorder. So we take the same exact reporter assay I showed you before. We take the MEF2C RNA sequence, tie it to luciferase, screen our compound library, and go through the same pathway. And since it is not going to be possible for us to do everything by ourselves, it means collaborating with people that do have iPSCs or model systems, animal models, and helping and working together to move forward collectively much more efficiently than just me doing it by myself.
All right. Thank you. Thank you, Dr. Buxbaum. That's it for the presenters. We do have food, so welcome to grab the food. We do also have a pre-recorded presentation that will be playing here shortly while I get food and socialize. Please, one last round of applause for the presenters today. Also, thank you all for participating and coming out today. It's pretty clear that we have a very strong community of researchers, doctors, professionals that are advancing research to help MEF2C, and also a strong community of parents here looking out for the well-being of their kids. So thank you, everyone.
Today to the MEF2C community. Thank you to Anne and the organizers. My name is Misha, and I'm a genetic counselor in the Simon Searchlight study. Today, I'll be structuring my talk with some general information at the very beginning, and I'll be diving deeper and deeper with examples of single nucleotide variants, copy number variants, and in the end, cover some general information about reading your genetics lab report. So genes are, the MEF2C genes are like recipes. This is what I think of when I think of genes. They are important instructions that are intentionally made that way to make a particular protein. So the MEF2C gene is important for the MEF2C protein. And I think of the protein as the product, the baked good, if you will. The gene's recipe needs to be followed in a really particular way so that the outcome, the product, is functional and the product you need. And so if you have too much sugar or not enough water in a recipe, it will end up messing up that product. It's the same way in a protein that if you have certain variations to that instruction, then it can ultimately disrupt the protein in some way. And proteins in this context are not actually proteins you eat. So I hope the example is not misleading. But proteins in this context are not the proteins you eat, but the proteins that are the building blocks of life. So in this presentation, I'm meaning proteins that are like enzymes, hormones, important for antibodies. It's important, for example, if you have a wound or you've cut yourself and your body will heal. Those are active proteins within the cells that will come together to help heal that wound. And so proteins are really the building blocks of functional life. Additionally, it's important to cover that genetic variations happen within every single one of us. I have genetic variants that are different than my parents, and they have genetic variations that are different than their parents. This happens in every single pregnancy. There is cellular machinery that is copying the genetic information to make sperm, to make eggs. And there are mistakes that happens. And that is a common occurrence literally in every single pregnancy. It's only when these genetic variations ultimately affect the proteins that how they function or where they're localized, or get rid of the protein entirely, that this can cause developmental or health challenges for a person. So if it's a problematic variation. But in general, most of us have these variations, and the most genetic variation that happens is actually not problematic to us in general. So now as we dive into an example, DNA, it's important to cover that DNA has 4 different letter codes. So you likely have seen this as well in your genetic lab report and the other talks that might have happened today, that there are 4 letter codes for DNA. So it is either A, T, G, or C. Within a gene, you'll have these letters in a particular order that read out and make this protein. And I'll dive more into that information. But let's dive into different examples of genetic variants. So single nucleotide variants can result in many different types of genetic variations. So one possible example of a single nucleotide variation is a missense variant. A single letter change that results in a protein amino acid change. And again, I'll dig deeper into this, but for now, what you can see is that in this MEF2C variation, and the C dot always means your DNA, the DNA code, and the P dot always means the protein code. But in the gene region of MEF2C, usually at site 113, there's a T. But in this person being sequenced, there's a C instead. And this single letter code means that instead of the protein usually having a leucine at site 38 of this resulting protein, there's a proline. So in this example, this would be it's expected in general that a protein would be produced, but the protein is altered in some way. The resulting function, so what happens when this amino acid is changed, is different in every single genetic community. So experts in your community would have a better understanding how the protein is altered when there is a proline instead of a leucine at this site. But this would be an example of a missense variant. A single nucleotide variant can also cause a nonsense variant. So in this particular case, we have a MEF2C genetic change at 860, where in the reference genome, so where the lab is using a comparison sequence of MEF2C, a DNA reference, that usually people have a C at this site. This person being sequenced has an A. This does not result in this case in a missense variant, but actually a nonsense variant. And I'll dig more into this as well. But here you can see that how this is represented is that normally at site 287 of the protein, there's a serine. But here, this little asterisk and it might show up on a genetics lab report as a TER or termination, or it will show up on a genetics lab report as an X. Those are all the same to say that this is a nonsense variant and this is a stop, that a stop was introduced in that protein. So another way to look at this type of or to call this type of protein variation is a protein truncating variant. So now I'm going to walk through more detail of how it goes from DNA to the protein. So to do that, you have to think of this intermediate molecule. So when there is a DNA sequence, a genetic sequence, a gene sequence, I mean, it's not that that recipe is cut out of the DNA and used once. No, just like a recipe card that you'll have that you'll come back to many, many, many times again and again. What you need is to read the DNA. So there's cellular machinery, as mentioned previously, that reads the DNA and it makes an intermediate molecule. It reads off the DNA and it makes what's called an RNA molecule. This is an intermediary molecule before making and translating, and that's the word in genetics, that going from RNA to protein is a translation. But there's an intermediate RNA molecule. And in the scientific world, it's actually very well known how this happens. And how this happens is that actually the RNA is read as a 3-letter code. This you can think of as a three-letter word in a sentence. And so it's read as the cat sat, for example, in this 3-letter sentence to make the protein come together. So in this example, let's walk through, in this particular molecule, you have a GCG. And the research and scientific community have broken down and created this rubric, this chart. It might look like a circle. It might look like it can come in different forms, but essentially they're all saying the exact same thing. Here is how the cell reads these 3-letter words in order to make the protein. And every single gene has a start, intermediate amino acid words, and a stop. In general, a general gene, that's how we can make genetic databases for humans, we have genetic databases for mice, where we can say, here are all the genes we're finding in the sequencing. We're not sure what all of these genes do. There's so many genes within every living organism, and researchers dig into and research what these genes might do, but we can tell what a gene is because we already know how to read the DNA. And so there's always a start, and there are stops. So that's important, and we'll cover that in the next example. But if you look at how to read this chart, we have this first letter word where it's G, C, G. So first position G, C, G. That gives you this alanine, this A in this example. So I'm not going to walk through the other ones. But what you do is you look for the first position, you find the second position in this category, and then you find the third position from this other side, and it leads you towards which amino acid would be read by the cell. So just highlighting again that there are 3-letter words for stop. There are 3-letter words to stop the production of a gene. And if a genetic variation introduces letters or shifts the letters in a way, it can result in a premature stop, a stop before the gene should have had one, or a protein truncating variant, as we discussed. So, more examples. This is a different type of single nucleotide variant. This is also resulting in a nonsense variant, but what happened in this case is there's a single DNA deletion of a letter, and that shifts the frame. That shifts the 3-letter word reading frame. So in the usual code, in the reference code that's being used in the lab, you would see in most of us in this region of MEF2C, AACT. But what's happened in the persons and the participants sample that we're using as an example here, this second A was deleted. It was removed. This is the genetic variation that happened in this person. And now the three-letter word is ACT in the DNA. What that results in the RNA code, and now I have to introduce a little more complex information, that the RNA made off this DNA, has another letter that it uses. So DNA is, A is always matched with T, and G is always matched with C, but RNA works a little bit differently. Whenever there's an A in the DNA, in the DNA code, a U will be placed. So A and U in the RNA. Whenever there's a G or a C, it will be, if it's a C, there'll be a G added in the RNA. If it's a G, it'll be a C added in the RNA. They're interchangeable. And whenever there's a T in the DNA code, there'll be an A placed. So as you can see, I've done the work of the transcription here, but going from DNA to RNA, this is the usual expected code. It would be UUGA, but this participant's mRNA would look UGA. So if you were to follow that, usually UUG would be UUG, would be leucine. But what happened with this particular single nucleotide variation is a UGA. So you can see still a U, but now we're over here, GA, which results in a stop codon, which results in a stop. So this is a protein truncating variant, but this is a single letter deletion. But we would still call this type of variation a nonsense variant. You could sometimes call it a loss of function variant. It's assumed that this type of variation would result in a loss of the protein function. Now there's another type of example that you can have with a single nucleotide change. I'm not going through exhaustive examples, but I'm just trying to showcase that there are so many different ways that the same type of genetic variation can result in different type of variants. And it always is very important to assess the variant and the variant in its particular context. So here we have now a frame shift variant. This is also a result from a single letter deletion. So here you have in the usual code a GAC, but the person is missing an A in this case. And reminding you of the DNA to RNA, I've done the work of adding in the participants' extended code as well as their mRNA code, just to show that in this case, the way that the genetic variant shifts the frame is an important feature of this type of genetic variation. So what has happened when there was this letter removal is that there's an insertion of several more amino acids, and they give you the number here. It's 8 amino acids were inserted that were now included that weren't normally the amino acids that should be there. So it shifts the frame. And then at the end of that 8, there's a nonsense variant. So again, you're still seeing that stop codon, that nonsense being introduced here. And I'll show you very specifically the amino acids that are now there. So normally a person will have a leucine at this site, and that's indicated here. The lab is saying that normally there's a leucine, which if you look at the chart, it would be a leucine. But what happens now with the shifted code is that you have these particular amino acids, and at the end of the 8th one, there's a stop included. So I've done the work. We won't walk through every single amino acid, but if you were to pause and look, you could see that every single there's all these 8 new amino acids, and then a stop is introduced. So you could call this type, and we would usually call this a frame shift variant. It does have a nonsense, a stop codon that is introduced. And it's also a protein truncating variant. It would also be assumed, although there's always exceptions in genetics, and there are general rules, but we always look at every single particular context for each person individually. But it would be assumed that this would be also a loss of function variant. So now to look at some copy number variants. So copy number variants, meaning that there is a large, and I'm using quotations because it's a loose description. I'll get more specific on the next slide, but a large amount of extra or missing genetic material, amount of genetic material on a chromosome. So this is just a cartoon of a chromosome, an example of where there might be a break. In a deletion context, it would make the chromosome shorter. In a duplication context, it would make the chromosome longer. More specifically, when we're wondering about a deletion, what would be considered a deletion variant, going by the scientific definition, it's defined by 50 base pairs to several thousands. So what that means is a deletion variant, when we think of one nucleotide being deleted, we would usually look at the protein consequence and call the variant by its protein, like we're saying, frame shift or nonsense. But when we think of a deletion variant or a copy number variant, it's 50 of those nucleotides in the DNA or more. And to dig deeper, if you were to have a copy number variant, you might have seen a particular number. So this is the cytogenetic location of the MEF2C gene. I'll go through what this means to say that this is an example of the chromosome 5. There is a P arm of every single chromosome. It's given to the arm of the chromosome that is shorter. So you can think of it as the petite, the small arm. And the Q arm is usually whichever arm of the chromosome is longer. The region where MEF2C is on the Q, the 5Q, you can see it here. And then this banding pattern represents how the chromosome is labeled. If you go to get a specific type of genetic testing and there's chromosome labeling, this would be a karyotype, then there's a specific chemical that's used that causes banding. And the banding is very characteristic for every single chromosome. It's how this long string of DNA is compacted into the structure so that it is really small and fits in our cells pretty easily. But this way that it's compacted is very patterned. And the banding, so you can see the band numbers, they increase, or they have a particular way that they are named. And so MEF2C is on this 5Q region 14.3 on chromosome 5. And to go further, whenever you get a result for a copy number variant, you will see these elements, these pieces of information on your genetic lab report. You'll see the reference. So this in orange is the reference that the lab used. The reference genetic information that each lab uses. And there's widely used ones, like this is a common one that's used, but it is updated regularly every few years, every 5 years or so. But as we sequence more and more people in general worldwide, we learn that what genetic variations are found in the general population and what are not. So as we're finding more and more genetic variation in the population, it's important to include that in the reference genome to say, here's what normal variation is, or here's what regular variation is. So every time you have these breakpoints, you have to look at it with the context of the reference genome that the lab used for their reference. So they will include that on the genetics lab report. You'll have the cytogenetic band information, as we said, what region is the gene on, and you'll be given the genomic coordinates. You'll be told, here is the first breakpoint end, here's the other breakpoint end. The last piece that you'll see is the information of, is it a deletion? is it a duplication? So ×1 means that you only have this person only has one copy of this genetic region. And so that means that they don't have two, which is the typical, most of us have 2 copies of genetic information, one from our mom, one from our dad. So on a genetics lab report, you actually won't see for the 5 chromosome, you won't see ×2 because any regular variation isn't being reported to you. But you will see ×3 if there's extra information on the 5 chromosome that might be problematic. Or ×1, which is a deletion. So this is all very important information for copy number variants. Finally, there was a question about how big is the MEF2C gene. So the MEF2C gene is over 160,000 base pairs. That includes this entire region of the gene. But this region of the gene within our DNA, within our genome, only has a subset of parts that are important for the protein coding. So here, if you go to the source I'm using is the UCSC genome browser, it's widely used but publicly available and free. If you were to go here, you could find the MEF2C gene, and the line across shows this region of the DNA. This is where the MEF2C is. But the vertical lines here are the regions of where the DNA is important for protein production. These pieces in between may or may not be important, but we would think of them as the introns or the in-between DNA that's holding the pieces of important information together. So when cellular machinery comes to make the RNA is made off of these particular vertical lines, and then the in-between is cut out. And so this is just the MEF2C gene region. So now I feel like I might be running out of time, but we'll touch briefly upon an example genetics lab report. So this is a cartoon example. It's written out. Not every genetic lab report will have the same pieces of information, but they often will have the items that I have itemized here. The layout also is very lab dependent, but in general, the results are presented on the first page of the potentially 9-10 page genetic lab report. Often your clinician, the evaluating provider, will provide some indication to the lab to gear their genetic sequencing. What type of genetic variation are we looking for? So they'll give some indications for testing. And then when the lab provides results, they will list the findings in often something like this way. So the result in this particular example is that this person has a MEF2C finding, and the lab is listing the reference that they used. So in this case, because it's a single nucleotide variation, they need the transcript information. So here is the transcript that was used that in order to look and reference the person's genetic variation. Often they will include, to help the providers, give some background information. So that's the next 2 pieces here. Because as we mentioned, there's more than 20,000 genes. It might not be that a provider knows every single genetic condition that's a possible result, but the lab will provide some background information. So is this gene characterized to be associated with a developmental condition or a genetic condition, yes or no? So is it a candidate gene, or is it uncharacterized, or is it characterized? We know that MEF2C is associated with a neurodevelopmental condition. Often they will include the OMIM name. So this is the OMIM. This is a online medical resource that clinicians and medical providers use widely, and genetic lab reports will also list any OMIM information on the genetic lab report. I've included that here. This is the reference number, and this is the name that's listed under the OMIM category for MEF2C. This piece is also more background information, usually for provider, but also, of course, understanding the genetic condition. It's the mode of inheritance. It's to say when there is a problematic genetic finding in this genetic condition, how does it present? The mode of inheritance in this case is not indicating how was this inherited. Sometimes I feel like that language can be a little misleading, but it is considered MEF2C is an autosomal dominant condition. So when there is one problematic genetic variation in this gene, then this does result often with a developmental or a genetic condition. Another type of mode of inheritance, for example, is autosomal recessive. So in those, you might have heard of cystic fibrosis, for example, you need 2 problematic variations in order to have the genetic condition. And so that mode of inheritance would be autosomal recessive. Where we start to get into the actual participant or person information is often in the next few pieces of information. So the genotype, how was this variant found? Was it homogeneous? Was there 2 copies? Or often in the case with MEF2C, you'll see 1 copy, you'll see heterogeneous. So now it's information that this person, they found 1 copy of this genetic variation and it was de novo. So in this example, there was likely parental testing. So that's the only way to determine if something or label something de novo, but was there a blood sample taken from mom and dad and were they tested negative? And so in this particular case, it was found de novo, arising new in that person. And what is the variation and the variant classification? So by now, you are likely familiar with looking at this. The C dot is the DNA letter change. So at site 43 in the DNA, usually there's a C. This person has a T. And it results in the protein change, the P dot of an arginine at amino acid 15 to cytosine. But now comes the important part of the variant classification. Is this problematic? Are we aware that it's associated with a neurodevelopmental condition? And yes, this is a pathogenic variation. Variant interpretation comes in 3 sort of main buckets of categories. So the benign or likely benign, those are the 2 subcategories in this bucket. But you wouldn't normally see benign or likely benign labeled on a genetics lab report because any benign or any non-problematic findings, they're not really reporting in general in this context. And so this would indicate that there's a genetic variation, but we see it in a population that doesn't have a neurodevelopmental condition or a problem with that feature being assessed. So we're not considering it a problematic variation. Then we have this likely pathogenic and pathogenic. As mentioned, there is a high level of certainty that this genetic variant has a consequence to a person's development and the resulting protein's function. The most annoying variant, but it is utterly common, is the variant of uncertain significance. So this is in the case where there might be some evidence to say that it could be a problem, but it's not highly confident that it's causing a problem, and it might not be a problem. So it's a variant of uncertain significance, or a VUS, or a VUS, depending on who a person's preference of talking about it, but they're all essentially the same thing, that more information is needed either over time, more scientific understanding, finding more people with a genetic condition that has the same genetic variant, or additional information like testing parents can sometimes help reclassify or help with the interpretation of a VUS. So the lab does interpretation, as you saw in the genetics lab report. There will be a level of interpretation. If, for example, a person has a VUS, and in general, the lab is often very cautious where they add something like this where clinical correlation is recommended. So that means to say that the evaluating provider, the person doing the genetic testing, needs to also critically consider the person in front of them and the genetic findings that are there. In order to sometimes help reclassify a VUS, that your provider could do that additional testing for the parents and help with criteria to understand if this is more likely to be a problem or not. So there is also a layer of the provider's role in interpreting a genetic variant. Thank you so much for your time. That's the end of my Genetics 101. Please send any questions my way. I'm sorry that I can't be there live in person today. And then you can also follow us on many of our social media channels for updates and other types of research information on our Facebook, on X, on YouTube with our informational videos, or on LinkedIn.